Hello everyone, this is Jack Golden, back at it again with Chapter 3.4 of Behavioral Neuroscience. So first we're going to talk about receptor molecules, recognize transmitter, and this is a continuation of 3.3. So ACH acts on at least four kinds of cholinergic receptors, nicotinic and muscarinic. These cells that use acetylcholine as their synaptic transmitter. Muscarinic organs work with the parasympathetic nervous system, nicotinic on muscles and automatic ganglia, mostly excitatory but also inhibitory. Otto found that the nervous system uses chemical signals as well as electric ones. Because of this, he won the Nobel Peace Prize in 1936. And in his test, he found that by stimulating a frog nerve, it released acetylcholine. We know that when there's changes, it could be due to seasonal variation, and many receptors show a daily variation of 50% or more. Upregulation increases the receptor number, and downregulation does the opposite. Transmitter bind to receptor gating channel. Ion receptor controls ion channels. Ligand gated ion channels that open and close in response to a particular chemical. Metabotropic receptors use G protein systems to alter the functioning of the postsynaptic cell. G proteins, okay, so proteins that reside next to the intracellular portion of a receptor and that are activated when the receptor binds an appropriate ligand on the extracellular surface. Okay, second messenger, slow-acting substance in the postsynaptic cell that amplifies the effect of synaptic activity and signals synaptic activity with postsynaptic cells. 80% of neurotransmitters and hormones activate cellular mechanisms through G proteins. The alpha subunit, part of G that migrates away within the cell and modulates activity of target molecule. The action of synaptic transmitter is stopped rapidly. Degradation is when chemicals break down the chemical breakdown of a neurotransmitter into inactive metabolites. Reuptake is when the released neurotransmitters are taken up and reused by the presynaptic part of the cell, stopping synaptic activity. Transporter receptor in the presynaptic membrane that recognizes neurotransmitters and sends them back. Several factors regulate neurotransmitter release. Autoreceptor, a receptor for a synaptic transmitter that is in the presynaptic membrane, telling the axon how much neurotransmitter has been released. Axodendritic is a synapse in which a presynaptic axon terminal synapses onto a dendrite of the postsynaptic neuron. Axosomatic, presynaptic axon terminal synapses onto the soma of the postsynaptic neuron. Axonic, all presynaptic neurons to regulate how much neurotransmitter will be released from the target terminal. Porosity, the axonal swelling from the neurotransmitter diffuse into a non-direct synapse. Non-direct synapse, pre- and postsynaptic cells are not in close opposition. Neurotransmitter is released by axonal velocities and diffuses away to affect wide region of tissue. Electrical synapse work with no time delay. Electrical synapse, region where neuromembranes are so close that they can change in potential and can flow from one to another without being translated into a chemical message. canaxon, a protein assembly that provides an open ion channel between two neurons, forming an electrical synapse between them. Gross electrical activity of the brain is readily detected. Electrical potentials large enough to detect at the surface of the skull. Two classes of these appear spontaneously without specific stimulation in those that are evoked by stimuli. EEG records brain potentials and waves, detects sleep cycles and seizures, and the condition of having seizures frequently is called epilepsy.